welcome. My name is Björn Högberg. I work at the Department of Medical Biochemistry and Biophysics. And today I'm going to talk about biology at the nanoscale and what we can learn using DNA origami. So I think that, in a sense, biology and all life itself is stored in chemical structure. So if you think about it, that's basically what biology is. It's knowledge stored into structure. And it can be complicated and convoluted, but appearing to be about something else often. But basically that's what it is. So it's knowledge, of course, about how to preserve this message. Self-replication, it's all about the preservation of previously known knowledge, from all the way to the smallest microorganism up to human society and the libraries and the internet. So what we have here in the background, I'm going to try to convince you that structure is important, and in particular at the nanoscale, which I think a lot of the important stuff in biology actually takes place. And what you have here in the background, I will try to explain this in further detail as we go through the talk. The bottom part here is a DNA origami nanostructure, and I will tell you what that is. It's made out of DNA, and the top part here is an antibody, an IgG, and of course the top molecule is designed by evolution and produced by nature, but the bottom part is actually quite artificial. It's something that we have designed to look like a rod, and we have designed it so that it would bind antibodies in a very specific fashion. And I will talk a little bit about that also later in my talk. But first, let me just give you a small introduction, what I think is important. So from early on in my studies, I have a PhD in physics. I always liked this quote from Richard Feynman: "What I cannot create, I do not understand." And I guess I like it so much because I really like to create and tinker with stuff, and just being, I think this quote sort of gives you the freedom to actually play around with things and be creative, and that could still be very important, because as you do that, you learn a lot about building stuff, and then you can actually use that to understand nature better. So is it actually true? I would argue that maybe it needs to be moderated a little bit. I think we cannot create galaxies, for example, but I think we can learn to understand galaxies. So maybe if we put it in this way, the things that we can create actually limits what we can understand. So the tools that we have at our disposal, that's the most important factor for any scientific study. So as an example for you, we had a German named Heinrich Geissler, and he invented a vacuum tube and a good vacuum pump in 1855. So he decided to capture vacuum inside a glass tube. So he pumped out this glass, he blew glass and pumped out the tube from air and created vacuum on Earth. I think he did that mostly because he thought that would be a cool thing to do. But it turned out that these tubes could be played with a lot, and Thomson used them almost 40 or more than 40 years later to discover the electron. And while playing with these tubes, Röntgen stumbles upon X-rays. And now, today, we have an X-ray diffraction image of what DNA actually looked like on a molecular scale. So normally, when I give my talks, I usually give them in such a way that I will tell everyone about the importance of a particular biological problem and the importance of understanding that, and then try to explain how we've gone about creating tools to understand these things. But I think for this type of talk, actually I would like to push the point that creating stuff is the important thing. So let's just dive right into what we can create. So any strand of letters can take form and structure, and in biology This is usually done by sequence-controlled polymers. So, just as my words that I'm speaking to you right now, they hopefully will change the structure of your brain, and at least for a few minutes before you forget about this talk. And structure in biology is also just a string of letters or words. And in DNA, it's defined by usually two strands, the blue one on the top and the red one on the bottom. And when they have the same type of words, they can meet each other and form this double helical structure. And in proteins, it's usually just one long string of letters. And when they interact, it interacts with itself to fold up into a three-dimensional structure. So making, the problem with protein structure is that we, as in the scientific community, we don't really know how this works. So this part here, we have very poor understanding of how that works. And we definitely can't just create new structures out of new words. Whereas with DNA, we have a very good understanding how this works. So if we have a specific type of sequence on the top and a specific type of sequence on the bottom, we know that it will always form this structure. And so this is something we take advantage of in DNA nanostructures. So if we have a sequence-controlled polymer like a protein, what happens is that some portion of the sequence along this length might interact to form a structure. So this protein is basically protein origami. And it's done in our cell continuously all the time, but our brains do not have enough knowledge to actually decipher what's going on. So one part of the problem is that we don't know exactly what parts of the protein chain that interact. So we could potentially, if we knew that typically the blue thing at the bottom would interact with the gray thing on the top, it should form this thing. But a lot of these things can interact with other parts, so let's say the blue part would also interact with the cyan part here in the middle, and then it could actually form this structure, and we have our tools to predict this are very, very limited. So with DNA, if we want to create something like this folded thing to the right here, we could actually design this interaction. This is something that we use a lot in my lab. So this part would be the long piece that we're trying to fold to the very left here. And then we can design this small piece in the middle here to interact. So the gray would interact with the gray and the blue would interact with the blue by hybridization or base pairing. And this would actually fold up the long thing like that. And this is what we do in this technique, which we call DNA origami. So this is DNA origami. This is an animation from one of our papers trying to describe how we have designed this icosahedral, it's actually much more faces than an icosahedra, out of about 200 DNA strands that self-assemble in a test tube. This technique was discovered in 2006 by Paul Rothermund at Caltech, but actually I would argue that in some form or another it has probably been around since the beginning of life, because this thing is like too easy. It works so well, so it must have been used by nature at some point. I don't have any proof of that, but that's something that I would like to prove in the future. So how does it work in more detail? Well, you start out with taking one long single-strand DNA molecule, and then you mix that with, like I told you, with a bunch of short single-stranded DNA molecules. And these are synthetic oligonucleotides that we design. So these are synthetic DNA, and the white thing here is actually something we take from nature, because we usually need a very long string of DNA, and that's very hard to produce chemically. So the long thing here, or the short ones, let's start with those. We call those staples. And as I said, they are synthetic DNA pieces that we just order. So you basically write the sequence in an Excel file and send it to the company that gives us the DNA in small test tubes back. And the long thing here we take from a virus that infects bacteria, a bacteriophage. So this usually from M13, MP18, and the reason we do this is purely because it's a nice way to get single-stranded DNA. We don't really care about the genetic information that this virus has used. In our purposes, it's just a long string of random words of DNA that we can use to address different parts here. So what we do is we design these interactions. Maybe I can use the. So basically, if you know the sequence of this long scaffold DNA here, let's say we know that we have C, C, C down here, and over here we have G, 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 or let's say A, 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 then we can design this yellow one to have G, G, G here and T, T, T here, so it would hybridize over here, and let's, just like I showed you before, it would staple the long single-stranded molecule together like this. And let's further assume that we designed the blue one exactly in the same way. We know the sequence of this long one, so we can just design this blue one to bind here, and this will also do that. And then it would start to build up a nanostructure made out of DNA. And if we add in small oligos to cover all the bases of this single-stranded DNA scaffold, eventually it will form these compact DNA nanostructures. And usually we depict these objects using cylinders as a representation of each DNA double helix. So we don't, I can just briefly mention the experimental work. So we don't really add the sequences in a complicated pattern. We just mix everything together and then heat it all up and cool it down in a normal thermocycler PCR machine. And then it forms these objects to a very high yield, usually above 90% yield of folded objects. So it's very easy. The design process is actually also quite easy nowadays. And this is something that we have worked on in my lab. So we have done artificial DNA origami in many forms. And here you see electron microscopy images of some of the structures that we have created. So on the top here, these are actually the things that we designed. So we developed an algorithm to basically just calculate the entire DNA design from a 3D object that we draw in a normal 3D software like Blender or Autodesk Maya, for example. So you basically just draw this shape, and then our algorithm will calculate the DNA design, which is depicted down here in the second row. And then the third to fifth row here, you have the electron microscopy images that we get after looking at these structures. And here the scale bars are 50 nanometers. So for this paper, of course, we designed a lot of structures, but actually this one, the last one here, we did not design. It's customary in computer science, for any one of you who had played with 3D meshes, that if you have an algorithm that's working on a 3D structure, you have to prove that it also works on something that is called the Stanford bunny. This is a 3D scan of a terracotta bunny made sometime in the 80s at Stanford. And it's used everywhere to prove that your algorithm works on a generalized 3D mesh. And so, of course, we had to prove that as well. So we downloaded this structure. This is a low polygon version of this Stanford bunny, and just fed it into our algorithm, and actually we got these nano bunnies. So we have this knowledge that the DNA origami are very easy to make. In my lab, I would say it takes less than a month for a master student to learn how it works and then design new structures and fold them and look at them in an electron microscope. And we can make almost any conceivable shape, as I am trying to show you here, very easily. And this makes me think again that surely nature should have used this kind of structural capability at some point to do something in nature. But we have yet, as I said, to find any natural DNA origamis, but I hope we will at some point. So why do we make these shapes? Is it useful to make a bunny out of DNA? And I think it is, because it really proves a point. And that is that these shapes and many other like them from this field proves the fact that DNA is actually one of nature's most exquisite building materials. And I don't think that was really appreciated before this field started to take off. And it proves the point that this technology could be very versatile and could be used to look at a lot of problems. And now I will sort of switch a little bit and focus on some of the examples that we have been doing in my lab to use DNA origami and use the precise structure capabilities of this technique. And in particular, I will look at two examples. And the first one is that we use, a lot of the time, we use DNA origami to position other molecules on the nanoscale. So not just DNA, we actually put proteins or small molecules on the origami using chemical conjugation of the proteins or small molecules to DNA. And when we do that, we can position these other molecules exactly in the pattern that we want. So here we try to use these structures to discover whether small separations between proteins can have a meaning for cell communication. And here we put efferent proteins on origami and then fed these patterns to of efferents to cells to see if differences in separation alone could influence the signaling of the F receptor. So it's been hypothesized for a long time that a lot of the mechanosensitive receptors in particular are dependent on clustering. So the signaling would vary and the response of the cell would vary if it encounters a certain pattern in its neighboring cell. And here, of course, we could use these structures instead of neighboring cells to actually very carefully measure the effect this activation has on a particular cell line. So we discovered that breast cancer cells, they did react differently if we fed them with efferents separated by 40 nanometers compared to efferents separated by 100 nanometers. And the F receptor activates to a higher degree. And here we basically use a technique called proximity ligation assay. You will just have to believe me when I say that these magenta dots here, they represent activated receptors. And we get much more of these dots when we use a 40 nanometer separation compared to 100 nanometer separation. And of course, the protein concentration, the total concentration of these efferent proteins is the same in all of these cases. And here below we plot the relative number of dots per cell in these cases, and we can indeed show that bringing these proteins closer together leads to a more efficient activation of the FA2 receptor. And also, it turns out that we, to some extent, actually change the behavior of these cells just by varying this very small, tiny nanoscale distance between proteins. So we looked at invasiveness, so basically you grow this breast cancer cell line on top of an artificial extracellular matrix, and then you see how these cells invade this matrix, and you measure how many of them have moved down the matrix, which is sort of also a measure of how invasive these types of cells would be in real disease. And we found that when feeding these cells with a pattern of 100 nanometers, sure, the invasiveness was depressed or inhibited, but even more so when we just move these proteins slightly closer together to 40 nanometers. So 40 nanometers is much more efficient at suppressing the invasiveness of these breast cancer cells compared to the 100 nanometers. So I would say that here we are using DNA origami to learn what I like to call the tactile alphabet of cells. By feeling their surroundings, cells react to how proteins are arranged on neighboring cells. But instead of sensing dots on the paper, of course, cells read nanoscale clustering of proteins in neighboring cells. So using this kind of technique to actually try to learn how cells communicate, I think is something that is very difficult to do with traditional methods. And this is an example where the precise positioning of DNA origami comes in really handy. So the other example I want to give before I finish off is that we've also recently used DNA origami in another nanoscale positioning experiment, and here we were interested in exactly how antibodies bind their targets. So antibodies are proteins that help eliminate disease, of course, from bodies, and they have two arms that can bind an antigen, for example, another protein or a virus. And if we show nanoscale patterns of antigens to antibodies, we would be able to learn exactly how far and how close the arms of the antibodies can bind. So I won't go into the technical details of this experiment, but basically what we could do, design different types of patterns of antigens, small molecule antigens that we positioned on top of this origami structures. And then we could do affinity measurements of these antibodies on top of the origamis, so they are binding to a specific pattern of antigens. And you can see here at the x-axis, we have the antigen separation in nanometers. And here on the top, you have the dissociation constant. So the higher the dissociation constant, the weaker the binding. So we see that for IgG, which is the most abundant antibody, IgG1 in our bodies, they have a very precise or very strong binding at exactly 60 nanometers. And it looks like IgM has, it looks like IgM, which is sort of the first response molecule of the immune system, has a very wide range of arm distances that it can tolerate. So IgM seems to be much more able in binding lots of different distances, whereas the IgGs are much more restricted in what type of distances they tolerate. So it's important to note here that the effective affinity, of course, goes down a lot when it's binding with both their arms. So up here, we hypothesize that they are only binding with one arm, and our experimental data corroborates this, and that below, in this region, they are actually binding with both their arms. So you would think that, of course, it's good to bind with both arms if you want to clear the body from pathogens, for example. So one thing we wondered is, why is then the IgM, which has a very broad range of arm angles, it seems, or very capable of binding lots of different distances, why is that molecule sort of ditched at the later stage of a disease when the body is producing mostly IgGs? So IgM is the earlier response and IgGs is the late response. So we have been hypothesizing that maybe this is to push the body into selectively trying to select antibodies with higher affinities in their individual arms instead of relying on two arms binding. So if you think about it, the combined affinity of an antibody would be both the individual arm binding affinity and also a factor from binding bivalently, or binding two arms at the same time. And if you have this combined affinity above the threshold level, let's say that this is the level of the selective pressure when the B cells are maturing in germinal centers, all of these clones would pass the selective pressure. But then it switches isotypes to IgGs, and then now some of these, which were relying a lot more on binding with two arms, are now actually falling below the selective pressure. And these would fail maturation, but only the ones that have actually really high affinity in individual arms would now pass the selection pressure. So this is something that we have been hypothesizing and are investigating further, and I think it's quite exciting. And a lot of these things, or all of the things that I'm showing you, but in particular this assay on the antibodies has not been possible before. So actually knowing this affinity and how it depends on the nanoscale separation between antigens, it's not known before. And again, I think here DNA origami has been a really valuable tool to actually precisely go in and look at these things. So I just want to end up with a little funny thing that we did in our lab when we had moved to the new research building in Biomedicum. I had asked my lab if one of them would like to design a nano biomedicum, and Erik Benson in my lab, he took on the challenge and designed this sort of the top view of our new research building. I don't know if you've been there. You should come and visit if you haven't. And designed the structure and took some pictures, and this was done in I think less than a week. And so we now have, of course, a lot of copies of this. If you want your own nano biomedicum, you're welcome to come by the lab and pick a small sample up. I think that the research in this field is, and in neighboring fields, it's just starting. So the funny thing about DNA nanotechnology is that it was all started as very curiosity-driven, a very curiosity-driven research field. So it was a lot of computer scientists actually who were thinking, so what is the most advanced nanostructure we can make, and how do we do it, and then they found DNA is really good for this, and then what is the most advanced complicated nanostructure we can build out of DNA. So, but just like with Geissler's glass vacuum tube, that eventually led to the discovery of the atomic structure of DNA, I think that it's really hard to predict exactly how this technique and other similar techniques will influence science, but it's very fun to play with DNA nanostructures, and the design that I've tried to show you, structure at the nanoscale, is of very big importance for biology, and we are just already beginning to discover things that would have been impossible to discover without the precise control that we now have. So again, what we can create really limits what we can understand. And with that, I would like to thank you for the attention, and my lab for all their great work, and some of my collaborators, in particular computer scientist Pekka Orponen in Aalto University, Anna Teixeira as our neighbor, and Jan Terje Andersen was collaborating with us for the antibody project. So, thank you for your attention.